All right, let's break down a topic that's not only fundamental to genetics, but also a huge deal for your NEET exam. We're talking about the Pachytene stage and the all-important process of crossing over.
So let's get right into it. The answer is tucked away inside our cells, in this incredible process that shuffles our genes around, creating all the amazing diversity we see. And look, getting a handle on this isn't just about acing biology class. It's a core concept that you absolutely need to know for your exam.
Okay, so where does this genetic shuffle actually happen? It all goes down during a special kind of cell division called meiosis. And we're going to zoom in even further into a really complex but super important phase called prophase 1. Think of this as the main prep stage. It's where all the magic of genetic mixing really begins. You know, prophase Y is actually so involved, it's broken down into 5 smaller substages. But don't worry, we're not covering all of them today. We're going straight to the star of the show, the most important one for genetic variation, where the real action, the recombination, happens. And that stage is called Pachytene.
All right, let's see how this all comes together. Pachytene. This is where it all unfolds. And I want you to remember, this is not a passive, boring stage. No way. It's super active, it's dynamic, it's the moment where the entire genetic recipe of an organism gets a major rewrite.
Okay, pay close attention here, because this slide has the exact keywords you've got to know for NEET. First up, you see those paired chromosomes, the bivalents? They're now clearly visible as a 4-part structure called a tetrad. Got it? Good. Now look at the next term, recombination nodules. This is the big one. These little nodules are the actual spots, the physical sites where the main event, crossing over, is going to happen. And what is that? It's the swapping of DNA between the non-sister chromatids.
And this diagram, it shows the process perfectly. Let's walk through it together. You've got the homologous pair, right? One from mom, one from dad. During Pachytene, their inside arms, the non-sister chromatids, they get tangled up. And at those points where they touch, that's where the recombination nodule does its job, helping them swap pieces of DNA. So what's the end result? Well, when these chromosomes eventually pull apart, they're not the same anymore. They're brand new, totally recombinant chromatids.
So for your exam, here's the deal. The most important thing is connecting the right words to the right stage. Seriously, the examiners love to ask about the sequence of these events, and if you can nail this down, it's an easy way to grab some points.
Okay, time to lock this into your memory. First fact, crossing over is the main reason for genetic variation in organisms that reproduce sexually. Got it? It's a huge deal. Second, here's a classic exam trap. They'll try to trick you with sister chromatids. But think about it. Why would you swap identical pieces of information? There's no point. The swap has to happen between non-sister chromatids to create something new. And hey, here's a little trick for you. P for Pachytene and P for passing genes. And please, please, please remember this trio. Pachytene, recombination nodules, crossing over. They always go together.
All right, let's put what we've learned to the test. This is a real question from a previous NEET paper, so it's a perfect way to see if you've got it.
You've totally got this. Just focus on that key phrase in the question, appearance of recombination nodules. Which stage was that? Go ahead, take a second and think about it.
And the correct answer is B, Pachytene. If you got that, awesome. Why? Because the appearance of recombination nodules is the defining feature of this stage. It's what Pachytene is all about. Remember, zygotene is when they pair up, and diplotene and diakinesis are when they start to separate later on. But the nodules, the sites for the swap, they show up right in Pachytene.
Okay, let's do a super quick 3-second recap. Ready? The stage is Pachytene. The site is recombination nodules. And the process is crossing over. Boom. That's the sequence you need to know.
And to wrap things up, here's a final thought for you to chew on. This whole gene shuffling thing, it's basically the engine of evolution. I mean, think about it. Without crossing over, genetic variation would just plummet, and a species would have a really hard time adapting to a changing world. It's just an amazing reminder of how a tiny microscopic event has massive consequences for all life on Earth. And that's it for this explainer. All the best with your studies.